Ich habe ja einen PC-Arbeitsplatz, also ich arbeite am Büro und vorwiegend am Computer, und mit einem Mal habe ich an einem Tag festgestellt, dass der Bildschirm kriselte. Es war nicht der Bildschirm, der kaputt war.
Imagine your eyes like a camera. Right at its center is a region called the macula, where everything comes into sharp focus. We use this area to read, see faces, and catch all visual details in our daily life. Now imagine losing your macular vision, no longer being able to see your friend's face across the room or read your favorite book. For millions of people, this becomes reality when they develop macular degeneration, the most common cause of vision loss.
Die Alltagsprobleme sind, dass ich zwar die Leute auf der anderen Straßenseite vielleicht noch sehe, dass da Leute sind, aber ich kann die Leute nicht mehr erkennen.
For people with Stargardt disease, macular vision fades away early in life. Unlike age-related macular degeneration that usually affects older adults, this genetic condition often strikes in teenage years or during early adulthood.
Stargardt disease is an inherited form of macular degeneration. In this disease, mutations affect the ABCA4 protein, which usually is part of a recycling system that helps to recycle byproducts that gradually build up when we see. Without this proper recycling system, toxic byproducts accumulate and damage the very cells that help us to see.
At the Institute of Ophthalmology Basel, clinicians and researchers work together to understand vision, its diseases, and to develop new therapies for vision loss. At IOB, we have a unique approach. We are not only relying on animal models, but also on human models of disease. And this ensures faster progression for therapy, and we also believe that we can provide better therapy for our patients.
Bis heute gibt es ja leider weltweit kein Medikament, keine Tablette, keine Therapie. Und so bin ich eben auch auf das IOB hier in Basel gestoßen, da es für meine Mutation hier ein Verfahren entwickelt wird, dass ich vielleicht doch noch mal eine Gentherapie bekomme. Dann wäre das schon für mich wie so 6 im Lotto.
Here at the IOB, we are working on a new promising treatment approach that fixes the faulty protein at its source, at the DNA level. Now the challenge is that the human genome contains 4 billion base pairs. Stargardt disease is caused by a mutation that affects a single base out of its 4 billion letters. Imagine you have to correct a typo in about 4,000 books containing 500 pages each. To do this, we're using a new technology called adenine base editing.
The adenine base editor is a protein complex made up of 2 key components: a DNA-binding protein that precisely targets the desired DNA sequence, and a therapeutic enzyme that converts an adenine base into a guanine base. The complex is guided to the ABCA4 site by a short RNA motif. By modifying this RNA motif, the base editor can be targeted to virtually any site in the DNA. Once it recognizes the mutation site, the base editor makes a precise correction. The other DNA strand is then repaired to match the corrected DNA strand. This one-time correction permanently changes the DNA sequence for the lifetime of the cell.
Previously, base editing was developed in cell lines, and it only worked in a petri dish or in mice. At the IOB, it took us 5 years to develop this technology specifically to the human retina and to the mutation that causes Stargardt disease. To package the base editor, we are using harmless viruses called adeno-associated viral vectors. This virus is like a crystal. It contains the coding sequence for the base editor. The virus enters cells like a Trojan horse, and once inside the cell, it quickly breaks down and it's eliminated from the body. However, the base editor protein is transcribed, and it will lead to a permanent change. Therefore, this therapy has to be only applied once, and it has an effect for a lifetime.
To develop base editing for Stargardt disease, we used laboratory-built human retinas, called retinal organoids, as well as real human retinas from organ donors kept in culture. Therefore, we could test our therapy in living human tissue.
With this technology, we achieved 80% correction rate. This means we can correct 4 out of 5 targeted photoreceptors in the retina. The therapy is meant to slow down or stop progression of the disease. Because we cannot regrow cells, our primary aim is to stabilize the condition and preserve vision for patients.
The IOB team will work further to bring this technology to the clinic. Next steps will include further optimizations of the technology and manufacturing of the gene therapy vector to finally allow application in patients. For patients like Andreas, this technology brings hope. The hope of preserving their vision. At the IOB, we are dedicated to turning this hope into reality, transforming groundbreaking research into therapies that can change the lives of patients.